Pharmaceutical Sciences in 2003 and completed his PhD in biochemistry in 2007 from the University of Porto in collaboration with the University of Montpellier. He moved to ICVS in 2014 to focus his research on the intricate relationship between cell metabolism, immunity, and infection, focused on parasites, okay, Leishmania. So these are circumstantial words. However, Silvestre is more than this. From my point of view, he is now, these days, a key research in the ICVS structure, and it's my belief that he'll be a key player in the future of the ICVS. So I'm honored to share some work with him. I'm really pleased for this to happen over the last few years. So without further ado, Silvestre, you can start, please.
Okay. Thank you, Jules, for the nice presentation. So I'm going to, and thank you all for coming today. I'm going to share with you a large part of the work that we do at ICVS that is focused on visceral leishmaniasis. And basically, yeah, you can hear me, right? Yeah, okay. And basically on these immunometabolic networks. So we try to understand how immune response function in a metabolic perspective, and try to give some clues on how we can, through the manipulation of the metabolism of immune cells, try to overcome some issues of visceral leishmaniasis. So let me just start by showing what is leishmaniasis. So we focus on one type of disease that is called the visceral one. There is a cutaneous and mucocutaneous. I'm going to focus on this visceral disease, which is a parasitic disease that causes visceral leishmaniasis, of course, in humans and in dogs, in veterinary disease as well. And it's caused by this protozoan parasite called Leishmania, that in visceral disease are Leishmania infantum and Leishmania donovani. So you can see a map here of the importance of this disease, and basically this is one of the neglected tropical diseases, and we have 20 of them. 12 of these neglected tropical diseases are actually parasitic ones, and Leishmaniasis, is among these neglected tropical diseases, is the second in mortality and 50 morbidity. So it affects mainly the visceral form, the South America, the East Africa, and the Asia as well. So we have the disease in Portugal, in south of Europe, mostly in a veterinary form, but we do have some cases of human disease as well, but is largely focused on these countries. So when I read the roadmap for neglected tropical disease that came out last year, so and despite the last 10 years of improvement, a lot of these neglected tropical diseases quite literally reduced the numbers of mortality and morbidity. So there is still a need in, and it is these two issues are among the high priority, as mentioned by WHO, to better understand the pathogenesis and the mechanisms of protection during human disease, mostly human disease, of course, and also to develop more effective and user-friendly treatments in order to try to eradicate this disease as it is planned during the next 10 years. Give you an overview of what happens in Leishmania. So Leishmania is a vector-borne disease. It is transmitted by this sandfly, which is a 2-3 millimeters sandfly, normally called phlebotomus or Lutzumia. It depends on part of the world that we are. And basically, when it bites, when it takes a blood mule and it bites a human or a dog, for example, it's going to insert these promastigote forms, these flagellated forms inside the dermis of these hosts. And these promastigote forms are quite rapidly phagocyted by macrophages and neutrophils mainly, and reside inside these cells, inside the phagolysosome of these cells as a mastigote form, which is the host-adapted form that resides inside these phagolysosomes. So basically, you can think that Leishmania affects or infects mostly macrophages and neutrophils in the first steps of the disease, But later on, because this induces a chronic disease, and when you take a look to several months or years after infection, the majority of infected cells are macrophages and dendritic cells, with a bit of neutrophils as well. And so the cycle continues because the mascots are going to burst infected cells and infect another ones, and it's going to continue the infection in the host. I must say as well that the main target organs of these parasitic protozoan, the spleen, the liver, so it induces the major symptoms of leishmaniasis, which is hepatosplenomegaly, but also it infects the bone marrow and other organs that I'm going to talk about later on. In terms of impact of this disease, so you can see some numbers here about the people at risk, mainly in the countries that I mentioned before, on continents I mentioned before. And although we for the past 10, 15 years, a lot of work has been done in trying to stop this type of not only leishmaniasis but neglected tropical diseases, we still have impressive numbers of infected persons. And I must say that one of the impacts of this disease, not only to kill the death of the infected people, so for example, visceral leishmaniasis is among the disease that is capable to induce HIV people, to induce AIDS in HIV people, but more importantly is the morbidity that induces in these people, especially visceral and cutaneous, because cutaneous leishmaniasis leads to a lot of disfiguring lesions as well. But more important than this, so we do not have a vaccine against human disease. We have a few on cutaneous, on canine leishmaniasis, but not on human disease. So the treatments are based on drugs, and we have a few of them, pentavalent antimonials, amphotericin B, the first oral drug that is methotrexate, and others that are still trying to understand if they work or not. And basically we have several issues with these drugs. The first one is the toxicity, which is common with drugs, but we have severe side effects on human treated with these drugs. But I will say that the main limitations of using these drugs are the resistant phenotypes that we have, not only with antimonials, but we have already resistance to methotrexate, for example, and paramamycin in several areas of the globe. Also, some of them have limited efficacy. For example, methotrexate has a partial efficacy on human disease and is very poor effective against canine disease, and it induces a lengthy hospitalization, which is a problem in this type of countries. Apart of the toxicity, resistance, and limited efficacy, one of the problems that it's very rare to have a complete cure. So we know that Leishmania can reside in some tissues. We still don't know very much where, but relapses cases are very common in patients with visceral leishmaniasis. In terms of the immune response to this, what we want when we want to eliminate the parasite. So this is a clear case of what we need is to activate macrophages, because I mentioned previously that macrophages are the most infected cells on human and canine hosts. And we have to induce macrophages to become activated to produce reactive oxygen and nitrogen species, so to induce nitric oxide or peroxide hydrogen, and to induce inflammatory cytokines. So to have this, we need to induce a response, an adaptive response of the type Th1 or Th17 as well. And particularly important is also the CD8 T cells, which have the capacity to become cytotoxic and to produce some molecules like granzyme and perforin that's going to kill the infected host, infected cell. So this is the way that we want to kill Leishmania. On the other side, we have the pathogen that tries to control the immune system, and also actually it forces the macrophages to become more so-called M2-like, so anti-inflammatory, and shifting the immune response towards a more IL-10 So in terms of, it is well known in the 90s, this Th1, Th2 dichotomy in terms of T cells, using a cutaneous strain was Leishmania major. But in terms of visceral disease, basically IL-10 is the major immunosuppressive cytokine that induces survival of the parasite. And on the other side, you want basically this, Th1 cells, CD8 activated cells, and to activate macrophages to kill the pathogens. So what is basically our perspective here? Our perspective is try to understand how this works, how this interaction between the macrophage and the pathogen host works in a metabolic point of view. So we focus on trying to elucidate the innate cell metabolism, so to find these pathways, so these players that we can manipulate in order to transform an anti-inflammatory macrophage in a more inflammatory macrophage and capable of killing the pathogen. So we use several tools. We use, of course, in vitro approaches with analyzes transcriptomic data, the metabolome of the cells, genome data as well. And in vivo, we have different types of models. So we use a lot at ICVS the neural model. I'm going to show in the next slides. We have different types of murine models in in regards of their susceptibility or resistance to the disease. And actually in collaboration with Paris, with Estaquis lab, we have developed a few years ago a non-human primate model of infection. So this gives us the possibility to bring these concepts of in vitro data to try to understand in terms of temporal and tissue compartmentalization. And it is, I must say that it's quite important because we know now that not all macrophages are the same. So you have different macrophages with different ontogenies. So it's very important to understand the interaction of Leishmania with these different types of macrophages inside the organs. And these two models, mainly the mouse at the ICVS, but also some data that we have with Estaquis lab in primates. And after knowing this, of course, we're going to try out every time to go to the clinical real relevance. So we have several collaborators in India that can give us some samples of human patients. And also we started very recently a collaboration here in Portugal with Utah University and in Brazil, in Bahia, trying to elucidate some of the data that we have in canine leishmaniasis. So of course, with the ultimate goal of trying to improve the treatment, basically trying to manipulate the immune response and try to have better outcome of the disease. So to give you some data that we have on the past few years, so we know, it is very well known that the metabolism of macrophage, for example, is linked with this function. So the majority of these studies were performed classically with cytokines that induce an anti-inflammatory stimuli or lipopolysaccharides that induce a pro-inflammatory stimuli. So very simple models that allowed us to understand how the metabolism of inflammatory cell works and the metabolism of anti-inflammatory cell works. And basically, I'm going to keep this simple, but if you have a pro-inflammatory that is capable of eliminate the pathogen, so the so-called M1-like pathogens, macrophages, so you have a cell that depends largely on glycolysis and the fermentation of pyruvate to lactate. On the other side, you have anti-inflammatory macrophages, M2 macrophages that depend mostly on the mitochondria and on the metabolism and the catabolism of lipids in order to perform the function. So we know that there is a clear association between several metabolic pathways and their function. So what happens in terms of our disease? So going back a few years ago, the first thing that we did at the moment was trying to understand in vitro what happens in terms when you put the Leishmania inside the macrophage. So Diana did this 5-6 years ago, and basically we observed that once we put on the same macrophage with the Leishmania, so the macrophage will respond mostly due to the activation of certain receptors like pathogen pattern recognition receptors. So it increases in the first hours the fermentation, it decreases the respiration of at the mitochondria, but this is quite controlling time. So this is a response of innate response of the macrophage that lasted around 10 to 14 hours. And the parasite is able to induce a metabolic switch that is going to be responsible for their persistence inside the macrophages. So one day and a half after infection in vitro, and we confirmed this in vivo, it is a different time points, but nevertheless, keep in mind that around 24 hours to 36 hours after infection, so we have a metabolic switch of the macrophages towards an increased mitochondrial respiration. And we observed that, in fact, this was associated with a disequilibrium in terms of these molecules, and an increase of the AMP-ATP ratio was observed during these 10 to 14 hours, which led us at that moment to think on AMPK, which is a protein that basically senses, is an energy sensor that senses this type of modulation. Story short, these AMPKs is a wide conserved protein among all the organisms, and basically what we observed at that moment was that, in fact, upon infection, Leishmania, only live Leishmania can induce this activation of AMPK, which will be responsible for increasing series of catabolic pathways and blocking all that is anabolic pathways, so trying to restore their energy of the cell. But more importantly for us, in terms of the immune function of the interaction between the host and the pathogen, was that, in fact, so as you can see here, AMPK is activated upon infection, was that, in fact, if you block AMPK, or if you block certain proteins that are responsible for the activation of AMPK, you can induce a phenotype of resistance both in vitro and in vivo. So overall, this first study led us to understand that, in fact, AMPK is a factor that is associated with susceptibility, that is manipulated by the parasite in the very beginning of the infection in order to induce a macrophage that was susceptible, and it was named to macrophages. And if you block this, you revert to M1 and you revert the infection. So at the moment, we thought this could be a good hypothesis as an immunotherapeutic approach. Unfortunately, what we observed was that this effect of AMPK was very restricted to the beginning of the infection. And if you take a look at chronic stages, we don't see any major effects. So knowing this, what we did was to, we changed the focus in trying to see factors, players that are associated with an inflammatory profile, with resistance to infection. And so, again, to make a long story short, George at the beginning, and then Ines during her PhD thesis, started to look, basically what we did was to infect macrophages that came from different types of mice, BALC associated with susceptibility to infection, C57 and 129, more associated with an intermediate or resistant phenotype. And among the genes and proteins that we look for, we observed that IF1 alpha, and this is the relative transcription levels of IF1 alpha during infection in vivo, we observed that these resistance or intermediate genes, mice, had higher levels of IF1 alpha when compared to susceptible mice. So we went to understand why this happens, and basically we take advantage for myeloid restricted IF1 alpha knockout mice. And what we observed was when we infect macrophages in vitro from these mice compared to wild type, we observed that we had a susceptible phenotype, so higher parasite burdens in the knockout compared to wild type. And actually, we had lower levels of TNF, IL-6, and higher levels of IL-10, meaning that in the absence of IF1 alpha, what we have is an anti-inflammatory macrophage is more prone to be infected with the pathogen. So we try to understand why this happens. So we know this gives us a perspective if IF1 alpha can have can be a protein related with resistance to infection. And so we try to understand why this happens. And to do that, and because IF1 alpha is actually a transcription factor, we make an RNA sequencing analysis of wild type and IF1 alpha knockout infected macrophages. And among the genes, among all what we have found, one thing has popped up, which was this BNP3. What we observed basically is that wild type macrophages upon infection upregulate this BNP3 protein, while knockout don't. And you can see this RNA sequencing data and qPCR made at the lab. So our conclusion at the moment was, okay, so you have an infection that induces E4 alpha, because I forgot to mention that E4 alpha is induced upon infection. So this E4 alpha is going to induce NIP3, and in the absence of E1 alpha, in the knockout, we don't have these phenotypes. So this led us to a large story, but basically what we found out was, in fact, as NIP3 has been previously mentioned as a protein that blocks mTOR signaling. And what we observed was that E1 alpha knockout mice, infected E1 alpha knockout macrophages, have displayed a higher level of mTOR activation. And this led, I don't have time to go through all the data, but basically the absence of IF led to mTOR activation, which led to, FRBP1C, which is a master regulator of lipogenesis. So what we observed at the end was that this factor that was associated with resistance to infection, if we remove it out of the equation, we induce the macrophage to produce much more lipids through the activity of these two enzymes. So we have lipid accumulation, as you can see here, measured by flow cytometry. We observe in the tissues as well, this oil red staining, that we have more lipids in the liver and the spleen of the infected knockout mice. And basically, this led us to understand that, in fact, this susceptibility was associated with increased accumulation of lipids. And if you block either FAS-N or the ACC, or this REBP, if you block this one or block this one, so blocking the lipid synthesis, you revert the susceptibility, just connecting that susceptibility in this knockout E1 alpha was associated with increased lipid accumulation. So actually we went further and we validated these on human samples. So E1 alpha has a lot of polymorphisms. We focus on one of them that is placed on the 3UTR of the gene, and this CTTT genotype actually is associated with lower levels of E1 alpha. And we observe basically the same. So these macrophages for the donors that have the CTTT genotype displayed lower levels of E1 alpha and higher levels of infected or number of parasites per macrophages. Mimicking a bit what we saw in the macrophages, and then as showing that the lower levels of E1 alpha or the absence is associated with higher parasite burden through the accumulation of lipids. To finish this part, this particular SNP is quite important for us. I think we need to go further and to validate in human samples, in patient samples, because their prevalence is quite high in the countries in East Asia, basically, in the countries where visceral leishmaniasis is more common. So basically, this is to close a bit this story. We have observed that E1 alpha is in fact a factor associated with resistance, and in their absence, or at least at lower levels of this protein, it unleashes a pathway that ends up by increasing synthesis of lipids and associates with susceptibility. So you can revert this if you block the lipid synthesis. So in this first part of the talk, we focus on some metabolic players. And I showed you that we focus mainly on one factor that associates with anti-inflammatory cytokines, so MPK, that is manipulated by the parasites to induce parasite survival. And on the other side, E1 alpha, this is also associated with the killing of the parasite. Actually, both of them interact with mTOR, and I must say that mTOR literature in terms of Leishmania infection is quite controversial. We found a bit of everything. mTOR activating associated with susceptibility, and mTOR activated associated with resistance. So we need to better understand the mTOR role in this context, and perhaps it is cell-specific, tissue-specific, or time-dependent as well. Entering in the metabolic pathways. So at the same time, or most recently, so we try to focus on the major pathways, so on glycolysis, on the amino acids, metabolism, and fatty acids. And I'm going to overview a bit of the data that we have so far. In terms of the metabolic pathways, we knew from the first study that in fact glycolysis is upregulated. I show you that with the work of Diana. Very recently, with these two collaborators from Germany, we actually published more or less the same in neutrophils. So it seems to be a similar mechanism. When Leishmania enters a macrophage or neutrophil, it induces increase on glycolysis. So our question here was at the beginning, and as a proof of concept so far, was is glycolysis important or not for the outcome of infection? So what we did was a simple experiment. So we took 2DG, which inhibits, actually it inhibits hexokinase, the first enzyme of glycolysis. And what we did was to give this drug before the infection, okay, just as a proof of concept. And we understood, or we saw at that moment, that if you block glycolysis previous to infection, 1 day previous to infection, we increase the susceptibility to the infection, both at the liver and the spleen and the bone marrow as well. So we have this data so far. So the question here that we pose ourselves is whether glycolysis can be or not a good pathway to target during infection. So we know that if it acts previously to infection, we can modulate the susceptibility, but we have no idea if we target glycolysis later on during infection, or when we target glycolysis, which type of cells are going to be affected by 2DG. So this is something, some questions that we have that we are going to try to answer in the next few months. In the meanwhile, and Carolina presented this very, very recently, in the meanwhile, another qPCR that we did, so Carolina found out that there was a signature of glutamine. So, okay, so we know that glycolysis is important, and macrophages are known to be quite avid consumers of glutamine as well. And we focus on this pathway. So the qPCR, we did a lot of qPCRs besides the RNA sequencing data, and we observed that in fact glutaminolysis was increased, so the usage of glutamine. And we checked in vitro, and we observed that infected cells have high glutamine consumption. So we try to modulate glutamine in vitro. It's not very easy because we have a lot of availability of glutamine in vitro, and the results were a bit problematic to understand. So we shift to in vivo. Let's try to understand what happens with the glutamine in vivo. And Carolina saw that during the infection, this is 1 month post-infection, for example, on the spleen, you have lower levels of glutamine on infected mice compared to non-infected mice. So this means that, in fact, I showed you previously that macrophages consume more glutamine, but I mentioned in the beginning that in order to have a protective response, we should not focus only on glutamine, but also on T cells, for example, Th1 and CD8 T cells. But this data shows us that lower levels of glutamine, meaning perhaps lower bioavailability of glutamine, can be present for the immune cells to control the infection. And what she did was basically to give more glutamine to mice. And actually, we didn't see anything different. So just giving glutamine doesn't solve the infection. But if you block glutaminolysis by inhibiting with BPTEs the first enzyme of the glutaminolysis, you induce susceptibility. So this comes in line with this data. So glutamine seems to be important. If you block glutamine usage by macrophages or T cells, I don't know, you increase susceptibility. So we reversed our thoughts, and we thought that perhaps glutamine can be useful if you give a drug that is going to kill the parasite. And that's exactly what we did. So we infected mice. We give metformin, which is an antiparasitic drug, which has a very short period of time. So we have around a 90% decrease in terms of parasite burden. And we give metformin in the presence of glutamine. So we added glutamine as a supplementation. And the effect was quite drastic. So we improved the efficacy of metformin around more than 100-fold. And so this was quite interesting to see. We published this last year. And what we want to know is why this happens. Why adding glutamine to metformin, we have an improved immune response, or at least improved elimination of the parasite. So we have performed a couple of experiments so far that put us on the road to understand what happens with with this synergic, or at least this butyrylfolin plus glutamine supplementation. The first clue came from when we characterized the immune response. We observed that among the cells that we checked on the spleen, CD8 T cells were the one that was increased in numbers, and particularly, I don't show it all the data, but particularly we observed that CD8 T cells have much more interferon gamma. Well, we found much more CD8 T cells producing interferon gamma, TNF as well. And so this pointed towards perhaps an important role of CD8 T cells in this context. It is not the first time that someone links glutamine with CD8 T cells. There is a couple of excellent papers on cancer showing that there are a role. Anyhow, in our case, what we did to prove or disprove that CD8 have a role was a very simple experiment. So we infected the mice as before. This is the same result as before, without drug, with drug, and with drug with glutamine. And we performed exactly the same experiment, but depleting CD8 T cells. So when we deplete CD8 T cells, so the first thing that you expect is without any treatment, we have an increase in parasite burden, just showing that CD8 are important for the control of diseases. But more importantly was this difference that we have it here, the difference of glutamine as fermentation. We lost it when we removed CD8 T cells. So this is the first proof that we have so far, and it's very recent data as well, actually, that the affected CD8, that the affected glutamine, by which glutamine works during infection, during treatment, sorry, is the effect on these CD8 T cells. Of course, we have to do CD4 T depletion, etc. But this point us clearly that CD8 T cells, perhaps because we increased bioavailability of glutamine in the spleen and giving the chances of the CD8 T cell to become active and to kill the infected cells. So this is something that we have very recently done, and we are going to continue to pursuing this mechanism. Okay, so so far in terms of the pathways, we show you that in fact, if you block glycolytic metabolism at the very beginning, you increase parasite survival. If you block glutamine metabolism, you increase parasite survival, meaning that in the beginning of the infection, glucose and glutamine is quite important for control of the infection. And more importantly, we show that supplementing with glutamine to an anti-parasitic drug, we decrease parasite survival and can be a good way to try to improve the treatment against Leishmania. So this opens the door to try to better understand how we can target the host metabolism. So I mentioned, for example, apart from glutamine, I mentioned glycolysis. We know that perhaps we need to improve glycolysis later on. One of the pathways by which we can improve glycolysis during infection is by targeting, for example, IFON alpha. IFON alpha is a resistant factor that induces glycolysis on macrophages, and there are a few drugs, for example, quercetin, DMOG, which are used already in vivo that can be used to target IFON alpha. So we will try in the next months, or at least perhaps years, to try to better understand if targeting host metabolism, if and how we can do it to improve current anti-Leishmania therapies. And the last part of my talk is relating to other potential targets. So what I've shown you so far is data that we have gathered mostly in the beginning of the infection. So we perform RNA sequencing a bit later on the infection. And if you focus not on the first 6 hours or 24 hours, but a bit late, so we lose a bit a signature for glycolysis and glutamine. Although what I said, it is valid because we prove it in vivo, but what I want to say is that if you look later on, we observed a signature on metabolism, but especially this is macrophages, human macrophages infected with Leishmania, but particularly metabolism of lipids and fatty acid metabolism, very much associated with lipid metabolism. So this is something that we had, that we got a couple of some months ago, and we are trying to so sequencing. She did the metabolomics, and the major hits that we observed was a lot of metabolism of lipids. I'm not going to talk about each, or at least not going to talk about this because we don't know what this means. This is just the data that we got. We feel that it's quite impressive to see that at later time points of infection, in vitro, of course, we have a very strong signature of lipid metabolism. We are very, very keen. I'm just going to mention glycerophospholipids. For example, sphingolipids and glycerophospholipids are constitute part of the membrane of the macrophages. And particularly this one we're mostly interested because we observed that inside this family, what we observed was an increase of a class of compounds called lysophospholipids. And we have a huge increase in this type of compounds are pretty well known to be secreted by immune cells, by macrophages, for example, in extracellular vesicles, and to have an immunosuppressive role at distance, for example, by blocking lymphocyte proliferation and other things. So we are trying that we are focusing right now to try to elucidate the role of this group of lipids in the context of Leishmania. But let me just come a little bit back. I showed in the beginning in D4 alpha story that the susceptibility associated with the absence of IF was due to an accumulation of lipids. So we have these fat macrophages. So we also know from other reports, and not only Leishmania but other pathogens, that in fact upon infection, macrophages tend to accumulate lipids. So it's something that is quite not common, but we see it a lot in different types of intracellular infections. So and because we have this signature of lipids in terms of metabolism, in terms of sequencing, we started to be quite curious what happens when you put lipids inside a macrophage. So we use a model where we take bone marrow cells, we differentiate them to macrophages in the absence of presence of adipose tissue. And when you put the presence of adipose tissue, you basically duplicate them even more the amount of intracellular lipids inside the macrophages. What we observed was that these macrophages in vitro are quite susceptible to infection, which associates the quantity of intracellular lipids with the susceptibility to infection. And one thing that is important, I show you only the in vitro data, but when you try to treat these macrophages compared to normal macrophages, we see, for example, that amphotericin B, so these fat macrophages, we don't have any anti-parasite activity. So amphotericin B do not work do not kill the parasites once they are inside macrophages that are full of lipids. We saw it in vitro, and we saw it at a lesser extent, of course, but in vivo as well. So this is another question that you have at the lab at the moment. So in terms of this lipid metabolism, and I'm going to focus now on lipid accumulation. What is the effect of this lipid accumulation on parasite survival? A few years ago, we did some electronic microscopy images, and you see that these lipids, these round dots here, are mostly colocalized with the phagolysosome. So you have here a parasite, you have here another parasite, and you have the lipids that colocalize with the parasite. So you have several possibilities, several hypotheses. So this is basically what we have submitted last year to the FCT and got funded, trying to elucidate this interaction between lipids and infection. So we hypothesize that we think that it can be an energy source, a nutrient source for the parasite, something that you have to prove. It's not proven yet, at least in terms of Leishmania. We think that it can work as a barrier to protect the phagolysosome to oxygen-reactive species and nitrogen species. This is a very simple experiment that we did with the normal macrophages and the fat macrophages, let me call that way. And we measure the peroxidation the lipid peroxidation, and we saw that was increased when we have more adipose tissue. If this means that it can serve as a barrier, we don't know, but it's certainly something that we will try to see. Of course, like I mentioned to you, this is going to influence membrane fluidity and phagocytosis, and even perhaps other things, because we have a huge modulation on these glycerophospholipids and others. And another thing that we think is this one: this adipose tissue can serve as a reservoir for infection. So we did some experiments trying to see if there is a reason, at least if this hypothesis can be correct or not. And we use a model where we fed the mice with normal diet or a diet high-fat diet, not to induce or put them a little bit overweight, as you can see here. So the average is not that huge, but they are overweight. And we infect the mice, and basically we observe the same phenotype that we did in vitro. So high-fat diet put the mice more susceptible to infection, both in the spleen. I didn't put here in the liver as well, in the bone marrow as well, and in the white adipose tissue as well. So you have more adipose tissue. It's normal that you have more parasite as well. What's important here? If you do the same experiment, but you treat the animals with amphotericin B, remember that I showed you previously that in vitro amphotericin B do not work in macrophages full with lipids. So if you treat mice, normal diet mice, you have a decrease of the parasite burden as expected. But if you do the same in high-fat diet mice, this decrease is more or less half in the spleen. But if you take a look to the adipose tissue, so the decrease on the parasite burden on normal mice is lower than in spleen, but still around twofold. But there is almost nothing happening with a high-fat diet, with animals fed with high-fat diet in the adipose tissue. So actually we have, I don't show it here, but we have some preliminary data, both from dogs infected with Leishmania, naturally infected dogs, and by non-human primates infected with Leishmania experimentally, that adipose tissue perhaps is a tissue where parasites can remain. And even though we can eliminate them from other important organs, like the spleen at the long course, they are going to reside in this tissue. So there's something that we need to prove, of course, but this data is is helping us in trying to put this hypothesis. So we think on the adipose tissue, among other things, as a potential reservoir for the parasites. So basically, this sums up a bit what we think about the adipose tissue. Not only that it can reside, but also the macrophages inside the adipose tissue are lipid-laden macrophages. So these adipose tissue macrophages are full of lipids that perhaps are the reason why the mastigotes can reside and can be maintained during chronicity and during infection. So what I tried to show you was our work during the last years is focused in trying to elucidate not only the players, but also the pathways manipulated by Leishmania during infection. So I show you to you at the beginning that AMPK is important as a susceptible protein, let's say, a susceptible associated protein, something that is manipulated by a parasite to persist. I show you as well that we understand now the role of E4 alpha as a resistance phenotype. We show that glutamine is important for the control as well as glucose. So these factors are important in defining resistance versus susceptibility to infection. And some of them, like glutamine, or even if we believe that it can be good immunotherapeutic targets, always in parallel with anti-leishmanial chemotherapy, of course. Also, we identify a SNP on the E4 alpha gene that associated with susceptibility. So this should be confirmed larger studies. And in terms of the metabolic pathways, like I said, a crucial role for glycolysis at the beginning, glutamine that can work as an adjuvant therapy, and there is a lot to know about lipid metabolism in this context. And I end up by acknowledging everybody in that contribute to these works in nationally at the ICVS, nationally, our international collaborators, and of course the funding that allow us to pursue these goals. Thank you very much. And if you have any questions,
okay, thank you, Ricardo. Very nice talk. So we have a question already from Claudia Nóbrega, and then Paulo Ludovico.
Hi, Ricardo. Very nice talk.
Hello.
Thank you very much. So I have a question regarding the knockout mice and also the SNP that you have identified. You saw that the macrophages from those mice and from those patients have increased parasitemia per macrophage. I was wondering whether those macrophages have increased capacity to phagocytose the parasite, or the parasite finds a more prone environment to proliferate.
We don't have any difference in terms of phagocytose. We've done it in several ways, but the percentage of phagocytosis in the beginning is the same. Actually, we don't see any difference. If the data that I show you, you have susceptibility around 4 or 5 days after infection. So the susceptibility is something that takes time. And that's why we show that is really associated with the capacity of this knockout to dysregulate lipid metabolism. So it increases lipids, increases synthesis of lipids, and this is the reason for the susceptibility. Okay.
Okay, Paula.
Okay. Hi, Ricardo. Very nice talk and very nice work. Thank you. Congratulations. I have several questions, but I think that they all go to one aspect of the work you presented, that is TOR activity and mTOR. How is mTOR here? And I would like you to comment on some aspects of mTOR. First, BNIP3 is known to be an activator, an indirect activator of mTOR. I think it is described that it goes through sometimes FOXO transcriptor factor. So one of the questions is whether or not you know if this interaction, if this activation is through FOXO transcription factor or not. Then another thing is that you mentioned all this data on glutamine, and glutamine is known to activate mTOR independent of RAG GTPases. So whether or not you have considered this. And finally, this is more related with the last results you presented. It is known that mTOR activation is quite dependent on the lysosomal surface availability, right? So when you have all of these lipids inside, you are increasing lysosomal surface, I guess. So whether or not you have this in consideration, and if so, lipids can be sensed by mTOR as well. I think that one of readouts could be the phosphatidic acid. I'm not sure, but there is a sensor that could be a readout for this sensing of lipids by mTOR. So as you see, all of this goes on mTOR. So I would like to have your comment on mTOR, on all the data that you presented. Thank you.
Thank you, Paul. That's an excellent question, because we made some, before I reply to your questions, saying that we did some preliminary experiments trying to see mTOR in different contexts. With lipids, for example, we did it. We didn't, still haven't take too many time to understand the role of mTOR in this context. So I cannot reply to you whether mTOR has a role in the fat macrophages, let me call it that, in that context, or for example, in the glutamine. We haven't checked. We haven't checked if mTOR has a role. What we know from the HIF1 alpha story was that, in fact, because these pathways, as you know, sometimes you can read it that it activates mTOR, sometimes you say it's inhibiting mTOR. Like I said in the presentation, one of the things I believe it is happening, it is cell specific. Let me say, is cell specific, time dependent, and perhaps organ specific. The other thing is that we need to improve our way to analyze this type of data, because we take 1 million cells in a well and we make a blot for mTOR, right? And we see mTOR activated. You don't know if the infected cells have mTOR operate or not. So all these technical difficulties can complicate the way we understand the role of mTOR. Like you said, it's true that MPK, E1 alpha all interacts with mTOR. That's why I said, if you take a look at the literature, and we and Carolina, we wrote a review very recently on that. You found 50% of the papers working on mTOR in the context of Leishmania saying mTOR is important for the protection, and 50% saying the opposite. So to close, I cannot answer your questions because we haven't done the experiments. All of what is said is true and is important, but I think we need to improve our way to analyze these cells, for example, by sorting cells.
Yeah, Ricardo. But the different availability of lysosomal surface could be underlying these different results that you have with mTOR activity. Once sometimes it gives parasite survival, and sometimes resistance, whatever. The availability of lysosome could be an important parameter to check.
But perhaps it's something that we can discuss later on, because I'm sure you know much more mTOR than me. To see if you can help us out to elucidate this, because we don't know really what happens with mTOR. Thank you. Margarita?
Okay, good. So we have another question from Patricia Monteiro. Patricia? Okay.
Thank you. Very, very nice and very clear, even for people outside the field, as it is my case. So if I understood correctly, the data from the F1 alpha knockout gives protection to these animals, right? And I imagine it's not easy to do a knockdown specific in macrophages of F1 alpha, but does that mean that if you do a bone marrow transplant from these knockouts to the infected mice, are you able to revert the infection, or this needs to be done before infection to protect macrophages from the parasite?
Well, the model that we have is a myeloid-derived knockout. So it targets, so you have knockout in myeloid cells, mostly macrophages, monocytes, and neutrophils as well. Okay, so we know that the phenotype that we observe, the susceptibility is associated with the absence of E1 alpha on those particular cells. Regarding the rest of the question, if you do, I'm sorry, I don't remember.
So if it's possible in that case, if you do a bone marrow transplant from the E1 alpha knockouts to the infected mice, is it possible to revert the infection, or this is only efficient if you do it before the infection of the parasite?
Well, actually, the E1 alpha are more susceptible. So the idea in that.
So they're worse, okay.
Yeah, but nevertheless, to answer your question, we tried to do that, actually. We tried to do it to show that, in fact, afterwards to sort the 45.2, so to sort only the infected, only the E1 alpha knockout mice. Well, what I want to say is that we tried to do that to show that not previously, previously to infection, we did it previous to infection to try to see if the knockout cells in vivo are more susceptible than the wild type cells in a chimera, right? So Inês tried to do it a couple of times. It didn't went well. So later on we let that experiment out. But I think it's a good experiment to do. The answer that's going to give me is the opposite that you were saying. So showing that in fact absence of E1 alpha even in an organism half competent are more susceptible. Something that perhaps we need to take a look again and try to do it.
Okay, thank you.
Okay, are there any more questions? Margarita?
Yeah.
Hi, Ricardo. Thanks so much for your presentation. Really cool. You present a lot of data on macrophages and metabolism, and of course, the one I liked the most was the single slide with the most interesting cells in the world, T cells. And I would like you to tell us a little bit more, because I don't know, probably I got a bit lost, because you show that if you do a certain treatment that change glucose metabolism, I don't remember exactly which one it was, that the effect, if you remove T cells, you don't see an effect anymore. So you conclude very well that T cells have an important part on this increased efficacy of the treatment.
Yeah.
So what I didn't understand is how can you conclude, and probably you can explain me better now, how can you conclude that this is an effect directly on the T cells and not on the activation of the macrophages that lead to the right activation of the T cells themselves? And then if you have time to tell us a bit more of like, metabolism is amazing, and we can really, and it's something new on the immunology, kind of new, and we really need to pay more attention to this. But how can we envisage that we can really manipulate metabolism to improve treatment? Because touching metabolism is something that I suppose is not really easy, not to have then a lot of side effects. So please, I'm sure you thought a lot about this.
Yeah, thank you, Margarita. Regarding the glutamine data, but what you said, I actually have two hypotheses. So what we know is that if you add glutamine during treatment with methotrexate, we improve the outcome. That's the first fact. Okay, that's clear. We published that. The second thing that we know is that in terms of the adaptive immune system, because we are looking at 1 month after infection, so you have, of course, the adaptive immune system working. We know that CD40, we didn't detect anything different in CD40 cells, but we saw that CD80 cells are more prone to at least produce interferon gamma and TNF. We haven't checked still granzyme and perforin. We are doing those experiments now. Carolina is doing now, but we know that. So that's the first hint that CD8 should be improved. When we depleted CD80 cells in this context, we lose the protection. So this is the second effect, second hint that led us to say glutamine is working on CD80 cells. Where we stand now, exactly on your question, is there a direct effect on CD80 cells, or instead is indirect by dendritic cells, by macrophage? We don't know, and it's something that Carolina is working on to try to understand that. That is, you got to the same question that we did. Regarding the therapy, so I understand very well. I thought on that, we've discussed already at the lab, how can we, if you touch metabolism, you are going to touch everything, okay? My perspective here is that if you add, if you use it like we are trying to use it, in concomitantly to chemotherapy. So the chemotherapy to Leishmania, for example, can last between 1 week and 1 month. It depends on the drug. So if you target on those periods and you boost the immune system, I don't think that, so the side effects that you're going to have in terms of on metabolism perhaps are limited. What we found out for several times, for example, and there are people from the cancer field here, when I say I give glutamine improve the resolution of infection, people from cancer say the opposite. I'm going to deplete glutamine in order to stop the tumor from growing, right? We are going to activate E1 alpha and we're going to eliminate the parasite. So Fatima is here and she knows they want to block E1 alpha to stop the tumor. So you have to be very careful. You cannot put this in terms of a chronic disease like 1 month, 1 year treatment. But I think to finish, if you restrict the treatment to very short periods of time, just to boost the immune system, you can improve it.
Thanks, Ricardo. Thanks so much.
Thank you.
I don't know if there are any more questions, but if that is the case, I have to leave to another meeting, so I can leave Tiago as host if anyone wants to keep discussing. Or
well, I can, if I may.
Okay, so I'm gonna just ask Tiago to host the. I don't know if I can do that.
Yeah, I'm already as a host, no problem. Thanks.
Okay, so I can leave. You don't need me. Okay, good. Bye-bye. Have a nice day.
Okay, Ricardo, very nice talk. Really interesting. Just very quickly, I was here wondering and thinking of drugs, and maybe I don't know different immune cells, but we know that metformin, for example, it actually stimulates glycolysis, and at the same time it improves the lipid profiles of people. So I don't know, maybe it would be worth trying metformin together with
in vitro, not in vivo. We tried in vitro, no difference at all. I was very surprised not to see anything there. Okay. But we've tried. Not in vivo. It's something perhaps we should do, because like you said, the effect is not only on macrophage, it's going to be on the immune cell as a whole, and perhaps you can see something else in later times of infection in vivo.
Yeah, because the effects of metformin will be different depending on the cells. And to answer your your comment about the cancer cells. Sometimes we also use metformin to try to kill cancer cells. So it really depends on what you do to the cancer cells. But of course, metabolism is a very good target. But sometimes we do the opposite. And actually, cancer cells are very flexible in terms of metabolism. So, and sometimes we actually have to target like both pathways of metabolism in order to kill the cancer cells or inhibit proliferation. So I don't know about this, if you would have the same kind of adaptation, let's say, when you use one drug in to try to target a pathway in metabolism, if there would be some compensation of a different pathway.
Yeah, that's always the same big question. We never know. We never know. Like Paulus asked. So there's these pathways are quite connected in several ways, so many ways. So you have to be very careful when you state something, when you test something. So, but that's it. We have to perhaps we have to do more in vivo studies in that way, because as I mentioned in the beginning, in vitro can be quite complicated due to the availability of different types of nutrients. Whereas the glutamine, we cannot modulate it in vitro because you have a lot of glutamine, same as glucose. When you go to those drugs that you mentioned, perhaps in vivo is the best way to take a look and to see all those perspectives. But well, there is still a long way to go. Anyway.
I was just wondering, like talking about metformin, if you have some idea that like diabetic people who use metformin, because metformin is one of the most used drugs to treat diabetes. Maybe like if you could have a cohort of patients, some of them who would take metformin and others who don't, but they would be different anyway. Maybe in terms of metabolism and body weight, I don't know.
I was seeing if Egidio is here or is not here. I know there's something going on with metformin and tuberculosis. I know that I think it induces susceptibility to infection. I'm not sure. That's why I was looking for Egidio, but he's not here anymore, or Gilles. But there is some studies, Margarita has left as well. Some studies with TB, at least that I know. In Leishmania or other parasite diseases, I never saw, but it's something that actually, well, to do that, I will say that we need to see something in vitro. More strong, because what we saw with metafusine in vitro was nothing at all. Nothing happens. Nothing in terms of parasite burden, in terms of immune function. We didn't see anything happen. But it's experiments that's easy to do, and perhaps to put not only Leishmania but other pathogens. If you get a cohort of metafusine treated individuals and untreated, to see if it increases susceptibility to different type of pathogens. Perhaps it's pathogen specific as well. I don't know.
Yeah. Okay. Thank you then, and congratulations again.
Thank you.
Does anyone have another question? No? Okay. So we'll just end this. Thank you, Ricardo, for a very good presentation.
Thank you very much. Thank you all for coming.
Yeah, thanks everyone to attend this Ciência Falada. We will be back in April 12th because of Easter. And well, we'll see you there. Have a nice week.